In 1978, a small piece of linen was clipped from one of the most examined objects in human history and sealed away for scientific study. Nearly 50 years later, researchers finally pulled the genetic code hiding inside that sample apart, piece by piece, and what they found wasn't one story. It was dozens of them, tangled together across 2,000 years, written into a single scrap of ancient cloth. This is not speculation, and it isn't a documentary reenactment dressed up as fact for a thumbnail. It's a peer-reviewed study published in Scientific Reports, one of the journals under Nature, and the findings genuinely surprised the researchers who spent years working on it. Stay with me, because the real DNA results pulled from the Shroud of Turin are stranger, considerably more nuanced, and ultimately more revealing than most of what gets said about this object online. Before we get into the genetics, it's worth understanding exactly what the Shroud of Turin actually is, because the object itself is already one of the most scrutinized pieces of cloth on the planet. It's a linen sheet, roughly 14 feet long, bearing the faint, straw-colored image of a man's body, front and back, along with what appear to be wounds consistent with a crucifixion, including marks around the wrists, the feet, and across the back and shoulders. Traditionally, it's considered by many to be the burial cloth of Jesus of Nazareth, kept today in the Cathedral of St. John the Baptist in Turin, Italy, where it's displayed to the public only on rare, specially designated occasions. Whether or not it's genuinely the burial cloth described in the Gospels has been debated for centuries, and this particular piece of research doesn't settle that question either way. What it does is something different and arguably more interesting. It reads the actual biological history written into the cloth's fibers, the traces of every plant, animal, microbe, and human being that ever came into contact with it, across 2,000 years of handling, veneration, and storage. The Shroud's documented history, at least the portion that can be traced with reasonable historical confidence, stretches back to at least the 1350s in France, when it first appears clearly in historical records connected to a French knight named Geoffroy de Charny. From there, it passed through a succession of noble European families before eventually settling in Turin in the late 16th century, where it has remained, with only brief exceptions during wartime relocations, ever since. Its earlier history, anything before that 14th century French record, remains considerably murkier and more contested, with some researchers proposing it may correspond to various historical relics described in earlier centuries under different names, though none of those proposed connections are considered definitively proven by mainstream historians. In 1978, during a major research campaign known as the Shroud of Turin Research Project, often abbreviated STURP, a team of American scientists was given rare direct access to the cloth itself for several intensive days of hands-on examination, an access level that has never been fully repeated since. Among the samples collected that year was a small fragment cut specifically from the edge, later used for radiocarbon dating, along with linen filaments and vacuum dust gathered from other sections of the fabric. Those original 1978 samples have since become the foundation for essentially every serious modern genetic study of the Shroud, including the research we're about to walk through, published just months ago by a team led by researchers Gianni Barcaccia at the University of Padua and Alessandro Achilli at the University of Pavia in Italy. What makes this particular study different from earlier attempts is the method. Older DNA studies of the Shroud, including a well-known 2015 study from some of the same research groups, relied on a technique called targeted PCR amplification, essentially searching for specific predetermined snippets of genetic code scientists already suspected might be present, then amplifying just those specific fragments enough to sequence and identify them. This new research took a considerably more thorough approach called metagenomics, sequencing every single fragment of genetic material present in the sample, regardless of what it turned out to be, without first assuming what should or shouldn't be there, and then matching each fragment against massive genetic databases afterward to figure out where it actually came from. Think of the difference this way. The older method is like searching a house for one specific type of object you already expect to find, checking drawers and cabinets you have good reason to suspect contain it. Metagenomics is like cataloging literally everything in the house, down to the dust in the corners, then figuring out afterward what each item actually is and where it came from. It's a slower, considerably more computationally demanding process, requiring powerful next-generation sequencing technology and extensive database comparison work, but it captures an enormously more complete picture of everything actually present in a sample, rather than only confirming or denying the presence of whatever researchers happen to already be looking for. This distinction matters enormously for a sample as old, as heavily handled, and as thoroughly contaminated as the Shroud almost certainly is. A targeted approach can only tell you whether specific genetic sequences are present or absent. A full metagenomic approach can potentially reveal entirely unexpected sources of DNA that nobody thought to specifically search for in the first place, which is exactly what happened here, and it's part of why this particular study generated so much fresh attention despite building directly on top of years of earlier research using the same original 1978 samples. And the picture that came back was genuinely complex. The researchers identified multiple distinct human mitochondrial DNA lineages present on the shroud, each one belonging to a different broad genetic population group, essentially different branches on the enormous family tree that traces back through maternal lineage across human history. One lineage, called K1A1B1A, turned out to match the mitochondrial genome of the actual scientist who collected the original 1978 samples, a completely expected and honestly reassuring finding, since it confirms the sequencing method was working correctly in picking up real, traceable human genetic material rather than noise or contamination artifacts nobody could explain or account for. It's actually a valuable built-in quality check for this kind of research, since researchers already knew in advance whose DNA the T particular signal should belong to, giving them a reliable way to confirm the overall sequencing pipeline was accurately identifying real genetic material. Beyond that expected match, though, researchers found additional lineages that weren't so easily explained. One called H2A2, matching the human mitochondrial reference sequence used as the baseline for comparison in genetics generally, a lineage common enough across parts of Europe that its presence alone doesn't point strongly in any single direction. Another called H1b, common throughout Western Eurasia more broadly, and a third called H33, a lineage the study specifically notes is prevalent in the Near East, and especially frequent among the Druze population, an ethno-religious group with deep historical roots across the Levant, sharing genetic ancestry with Jewish, Cypriot, Palestinian, and Syrian populations, who have intermixed across that region for thousands of years. That particular lineage is the one the Vatican's own statement on the research specifically highlighted, since it aligns most directly with a Near Eastern chapter of the cloth's proposed historical journey. Beyond the human genetic material, the metagenomic sequencing also reconstructed an entire microbiome living on the cloth's surface, a rich mixture of microbes commonly found on human skin, along with archaeal organisms specifically adapted to extremely high salinity environments, the kind of conditions you'd find somewhere like the Dead Sea region, plus various fungi, including several types of mold that would be expected to accumulate on any organic textile stored for centuries in varying humidity conditions. Researchers also detected DNA from Mediterranean endemic red coral, along with genetic traces from a range of cultivated plants, including carrot, wheat, corn, bananas, and peanuts, and from domesticated animals as well, likely reflecting everything from historical handling practices to environmental exposure during the cloth's various documented relocations across Europe. Put together, this genetic snapshot paints a picture of an object that spent an enormous amount of time exposed to a Mediterranean environment, handled repeatedly by different people in different places across a very long stretch of history, its fibers slowly accumulating a kind of biological diary that modern sequencing technology is only now capable of reading back out. Now, here's where the story gets genuinely surprising, and it's the detail that made headlines when the study came out. According to the research team's analysis, nearly 40% of the human genetic material recovered from the shroud traced back specifically to lineages associated with the Indian subcontinent, a proportion large enough that it couldn't reasonably be dismissed as a handful of stray, isolated contamination events. That's not a small or easily dismissed statistical blip. It's a substantial portion of the total human genetic signal detected across the entire sample, combined with earlier findings from the same broader research program, including analysis of plant DNA that pointed toward possible origins in the historic Indus Valley region, an area whose textile traditions stretch back thousands of years and were historically renowned for producing exceptionally fine linen and cotton fabrics. The researchers suggested this pattern might indicate the linen itself, the actual woven cloth, could have originally been produced somewhere in India, before eventually making its way westward toward the Middle East and, much later, Europe, potentially traveling along established ancient trade routes that connected South Asia to the Mediterranean world for centuries before the cloth's documented European history even begins. The Vatican itself publicly commented on these findings, noting that the new research appeared to confirm the shroud's passage through the Middle East, and specifically highlighted the detection of that H33 lineage tied to Near Eastern populations, along with the identification of microorganisms adapted to thrive in extremely salty environments similar to the Dead Sea. Taken together, the Vatican's statement framed this as broadly consistent with a Middle Eastern chapter in the cloth's long history, alongside the newly proposed Indian connection tied to its possible textile origin. But here's the part that matters enormously, and it's exactly where a lot of the more dramatic headlines and video titles floating around online tend to lose the plot entirely. This DNA evidence does not, and cannot, confirm who the shroud actually belonged to, nor settle the centuries-old debate over its authenticity. In fact, several genetics experts who reviewed this research after publication were considerably more cautious about its conclusions than the initial headline suggested, urging readers and viewers to treat the more sweeping claims with real scientific skepticism. Christina Warriner, an anthropologist at Harvard University who specializes in studying ancient microbiomes, reviewed the findings and noted that while the human and microbial results in this particular study are likely trustworthy, from a technical standpoint, the plant and animal identifications came from a different, less reliable matching method, one known to handle plant and animal genetic data considerably less accurately than it handles human or microbial sequences. That distinction matters enormously because a fair amount of the more sensational interpretation of this study leaned heavily on those specific plant and animal findings, the ones flagged as scientifically shakier ground to begin with, rather than on the more solid human and microbial data the researchers themselves expressed greater confidence in. There's an even more fundamental problem underlying literally every DNA study ever conducted on the Shroud, and it's one the researchers themselves are careful to acknowledge directly in their own published work. This object has been touched, kissed, carried, displayed, and venerated by an enormous number of people across roughly 2,000 years of documented and undocumented history, passing through the hands of knights, nobility, clergy, scientists, and millions of ordinary pilgrims over the centuries. Every single one of those interactions had the potential to leave behind trace genetic material, skin cells, saliva, sweat, hair, pollen carried in on clothing, dust from wherever the cloth happened to be stored or transported at the time. What that means, in plain terms, is that the DNA recovered from the Shroud isn't a clean, isolated sample from one original source, preserved untouched in a sealed environment. It's a genuinely tangled, layered mixture accumulated from centuries of contact, and untangling exactly which genetic traces belong to the cloth's original historical context versus which ones were left behind by a pilgrim praying over it in the year 1600, or a researcher handling it during a scientific examination in the 1970s, is extraordinarily difficult, and in many cases may simply be impossible to determine with full confidence using genetic evidence alone, no matter how sophisticated the sequencing technology involved actually becomes. This challenge isn't unique to the Shroud either, though it's particularly pronounced. Here, given the object's extraordinary level of historical handling and veneration across so many centuries and so many different cultures, any ancient textile or artifact that's been extensively touched, displayed, or richly handled over long stretches of time faces this exact same fundamental problem when researchers attempt genetic analysis, from Egyptian burial wrappings to medieval religious relics housed in cathedrals across Europe. Archaeologists and ancient DNA specialists refer to this general issue as the contamination problem, and it's one of the most persistent, genuinely difficult challenges in the entire field of ancient genomics, regardless of what specific object or culture happens to be under study at any given time. The researchers themselves describe this directly in their paper, writing that their findings, combined with earlier research from the same group, provide valuable insight into the geographic origins of the many different T individuals who interacted with the Shroud throughout its long historical journey, across multiple regions, populations, and eras. Notice the specific language there. Individuals, plural, interacted with, not necessarily originated from. This is a study about accumulated human contact over centuries, not a single, clean genetic fingerprint pointing to one specific person or one specific moment in history. This particular nuance is exactly why coverage of the study has varied so dramatically depending on the source. Some outlets ran with the more sensational angle, framing the Indian genetic connection as potential evidence that the cloth itself might trace back to the ancient Indus Valley civilization, occasionally implying this could support a much older origin story for the object than mainstream historical dating currently suggests. Other, more scientifically cautious outlets pushed back hard against that framing. One widely read piece in Scientific American ran with a direct headline stating that the DNA analysis doesn't actually reveal the relic's true origins, quoting outside experts who stressed that this research, while genuinely interesting from a pure genetic standpoint, doesn't meaningfully resolve the core historical and religious debate that surrounded this object for centuries. It's also worth noting explicitly that at the time this particular study was first published, it had not yet gone through formal peer review, a detail multiple outside scientists specifically flagged as an important caveat limiting exactly how much weight should be placed on its more dramatic conclusions until the full peer review process had actually run its course. A subsequent related paper from the same broader research group was later published through Scientific Reports, which does carry full peer review, but even that version maintains the same fundamental limitation. This is a study of accumulated contact and contamination, not a single isolated genetic origin story. It's worth briefly touching on the radiocarbon dating question too, since it inevitably comes up whenever the Shroud's authenticity is discussed. And it provides useful context for understanding what this new DNA research does and doesn't add to the broader picture. Back in 1988, 3 independent laboratories, working in Oxford, Zurich, and Arizona, each performed radiocarbon dating on a separate, small sample cut from the cloth, and all 3 arrived at strikingly consistent results, placing the linen's origin somewhere in the medieval period, roughly between the years 1260 and 1390, a range that notably excludes the 1st century when the historical Jesus of Nazareth would have lived. That dating result has remained deeply controversial ever since, with some researchers arguing the specific sample tested may have been contaminated by centuries of repairs and handling, since it was taken from a corner of the cloth that historical records suggest was patched or rewoven at some point, possibly following fire damage the shroud is known to have suffered in 1532. Other scientists maintain the original 1988 dating results are sound, methodologically, rigorous and reliable, and that proposed contamination explanations haven't held up well under closer scrutiny. This particular DNA study doesn't directly resolve that specific dating controversy either way, since genetic material and radiocarbon dating are measuring 2 completely different things. One identifies biological origin and historical human contact. The other measures the physical age of the cellulose fibers making up the linen itself, based on the decay rate of a specific radioactive isotope of carbon. So what does all of this actually add up to, once you set aside the more sensational framing? A genuinely fascinating, technically impressive piece of modern genetic research managed to pull a remarkably detailed, biological history out of a single small fragment of ancient cloth, identifying human genetic lineages tracing back to Western Europe, the Near East, the Arabian Peninsula, and the Indian subcontinent, alongside plant, animal, and microbial traces, consistent with extensive travel and handling across the Mediterranean world. That's a legitimately remarkable scientific achievement in its own right, regardless of what conclusions anyone ultimately draws about the Shroud's religious significance. It demonstrates just how much biological information can still be extracted from an object that's been examined, poked, and studied for well over a century, using genetic sequencing technology that simply didn't exist for most of that examination history. What it doesn't do, despite what more dramatic headlines might suggest, is prove or disprove whether the Shroud actually wrapped the body of Jesus of Nazareth. It doesn't confirm a specific point of origin with full scientific certainty, and it doesn't overturn or replace the earlier radiocarbon dating results, controversial as those results remain among researchers on both sides of the broader debate. What it does provide is a genuinely rich, if inevitably tangled, genetic record of centuries of human contact with one of history's most examined and most debated religious artifacts, a record researchers will likely keep working through, refining, and reinterpreting for years to come as sequencing technology continues to improve. There's something worth sitting with in that conclusion, honestly. The real scientific story here isn't a single dramatic revelation that instantly settles a 2,000-year-old mystery, the kind of tidy, cinematic ending a video title might promise. It's something quieter and, in its own way, more genuinely fascinating. A scrap of linen that has passed through countless hands across 2 millennia, silently accumulating a layered genetic record of everyone who ever touched it, prayed over it, or carried it from one place to another through wars, fires, relocations, and centuries of religious devotion. Modern genomics is only now developing the tools sophisticated enough to start reading even a fraction of that accumulated history back out of the cloth. And the researchers themselves are the first to admit there's still an enormous amount of careful, patient work left before anyone can confidently separate the Shroud's original story from the 2,000 years of human contact layered on top of it, one careful sequencing run at a time. If deep dives like this, ones that walk through the actual peer-reviewed science behind a famous mystery instead of settling for a sensationalized headline, are exactly the kind of content you come here for. Make sure you're subscribed to this channel with notifications turned on, because researchers are continuing to study these samples. And as new results come in, we'll be here to break down exactly what the data does and doesn't actually tell us.